So welcome to the NDSU Public Health Seminar series. We've been doing these roughly once a month for this year and had some outstanding presentations. And today is another example of such. Today we have Dr. Rick Jansen presenting to us on the topic of multi-omics analysis of public health data on pancreatic cancer. Dr. Jansen has been here at NDSU since 2015 as a colleague and friend of mine. He's an outstanding informatics expert, and today we're having a chance to hear from some of his research to that end. Dr. Jansen earned a Master's of Science and a PhD at the University of Minnesota in Environmental Health Sciences with minors in epidemiology and biostats, and also has completed 2 different postdocs in cancer genetic epidemiology. So he's well prepared to speak to us today on this topic. Dr. Jansen's research interests include today's topic in genomics, epidemiology, cancer epidemiology, epidemiologic methods, spatial epidemiology to model infectious disease transmission and dietary intake assessment. He teaches environmental health and upper level epidemiology courses in our program, and we're thrilled to have him presenting today. So take it away. Oh, I should probably clarify. Please feel free to enter questions in the chat box. And Dr. Andrea Huseth will be managing our Q&A time today. So Rick, you can go ahead and share your screen and start your presentation.
Great. Thank you, Dr. Strand. Let me share my screen.
There we go. Looks good.
All right. So as Dr. Strand let you know, I'll talk a little bit about multi-omics analysis approach to pancreatic cancer and dive in a little bit deeper into what is genetic aspects that influence pancreatic cancer, what are epigenetic aspects that influence pancreatic cancer, and then thinking about how risk factors also play a role and how can we combine those different data sets together in a way that can help inform the underlying biological mechanisms that might be at play. So I first wanted to start off by just giving a little bit more of a background. As Dr. Strand mentioned, I got my master's and PhD at the University of Minnesota in Environmental Health Sciences. I got minors in epidemiology and biostats, and my dissertation focused on trying to understand how case ascertainment bias influences the way that we sample and select participants into our observational epi-type class studies. And so this happens anytime there's early detection used to detect. So those screening mechanisms or other factors that lead individuals to go to the clinic and get diagnosed at an earlier date than those people that don't go to the clinic and get diagnosed earlier. And so that automatically will have an effect on who's in your study, who's classified as a case versus a control, and also has a direct impact on survival estimates within the study. I see that my slide messed up a little bit here, but then I did a couple postdocs, one at Mayo Clinic, focused in genetic epidemiology of pancreatic cancer, doing some food frequency analysis questionnaire for understanding how dietary factors are associated with pancreatic cancer. And then another postdoc at the University of Chicago. There it was more where I got exposure to the bioinformatics type of analysis approach and trying to understand some epigenetic related factors that were related to aging in a population in Bangladesh. So since coming to NDSU in 2015, I've worked in several different areas. So if we start up at the upper left-hand corner, my work work focused in cancer, both breast cancer and pancreatic cancer, and collaborators at Eastern Virginia Medical College and also Mayo Clinic Health System. And then moving down, sort of using my bioinformatics background to take a look at these multi-omics data sets and think about how we can analyze those and use those to understand a little bit more the complex disease and biological mechanisms at play with different types of cancer. And then in the bottom right-hand corner, using my biostatistics background and developing those mathematical models related to case ascertainment bias. As we move up, using more of my environmental health background to understand how environmental conditions could influence diseases like obesity in children or arsenic exposure in Bangladesh. And then some cardiovascular statistical work related to some of those features at play within North Dakota. So for today's talk, we'll start off with a background in pancreatic cancer, move on to discussing some of the known genetic factors that influence the disease, and also how we can incorporate epigenetics into the picture as well to further our understanding. Look at a background of bioinformatics approaches that are currently used in the study of cancer, and then going through some of the results that I've produced using funding, a COBRI funding mechanism through the university here at NDSU, and then future pancreatic cancer research directions. All right, so if we ask the question if pancreatic cancer is the worst cancer type, it really depends on how you're going to define that. So we know that it affects the pancreas. The pancreas is deep within the abdomen, and it's difficult to visually detect or image in the clinic any alterations that might be within the pancreas. And so oftentimes there's going to be a delay in diagnosis until pancreatic cancer is at a later stage. And so oftentimes there's going to be more severe symptoms associated with the cancer. And being at a late stage, as is true with most cancers, the cancer itself isn't going to be as responsive to existing therapies and treatments that we can use in the clinic, because there are so many different types of cancer cells there at that point. And also the potential to have metastases, so the cancer cells moving to different organs, to lymphatic tissue around the body. All right, I don't know why this one isn't showing up. Maybe I shared the wrong presentation. Let me stop sharing this one and jump over to the other one. Sorry about that. There we go. All right, can you all see this? All right. So
Yep, looks good.
Again, it depends on the context and the metrics you're going to use. So are you looking at incidence? Are you looking at survival? Are you looking at, here, let me back up one more slide. Are you looking at which gender are you looking at? And so that will all influence and help you decide whether pancreatic cancer is could be classified as the worst type of cancer. So if we take a look at the table here on the right-hand side, we see that in terms of all different types of cancers in the United States, pancreatic cancer would be 11th if we were looking at incidence, and it would be 3rd if we were looking at deaths. And so actually with some updated numbers, it would actually be the second cancer type in terms of the number of deaths it causes in the United States at this point. We also can see that there is a little bit of a difference in terms of cancer types and how they affect the different genders. So taking that into account as well. And then if we would use a metric to ask individuals how concerned they were about different cancers, we would still have a different order to the way that the different cancers would line up in terms of those that would be considered most significant and those that would be considered least significant. So if we look at pancreatic cancer itself, most of the time, the cancer is going to be affecting the ductal system, so right in the middle of the pancreas, again, potentially leading to it being harder to detect using CT imaging or MRI imaging. So we see that from this table here, over 90% of the cancers are going to be classified as adenocarcinoma, and endocrine just means that it's going to be in the ductal system, and neuroendocrine means that it's going to be outside of that ductal system. The neuroendocrine cells within the pancreas are going to be the cells that are responsible for secreting the digestive juices that help with digestion into the intestines. All right, so if we also, I mentioned a little bit about it being the late stage causes a lot of the issues and the poor survival in pancreatic cancer. So if the cancer was actually diagnosed earlier, it's thought that it would be easier to treat, that the cancer itself would be more responsive to those treatments. So one thing we run into as epidemiologists is that we only can do observational studies. So we can only know that somebody has pancreatic cancer when they've been diagnosed. So if most of the cases are diagnosed earlier or later, at a later stage, that's where we have most of the information. Those individuals that are actually detected earlier aren't necessarily representative of all people that could potentially be detected early. So for some reason, their cancer is symptomatic, or for some reason, somebody's looking at their pancreas. So that leads to the discovery of an earlier stage pancreatic cancer. And so those that are actually diagnosed in the clinic aren't necessarily representative of the entire population had they been diagnosed early on. And that's sort of what the lead time and survival time is showing in the upper right-hand corner here. The fact that if you're clinically diagnosed, you're going to be automatically diagnosed earlier. And so if you're using a screening test, or if it's caught for another reason, that's not directly related to the cancer itself. And so that's going to extend your survival time. So if we say that those detected earlier have a longer survival time, well, that's a given because they've been detected at an earlier stage, but that doesn't necessarily mean had we detected those individuals earlier that were diagnosed at, say, stage 4, that they would have an improved survival time. But that's really the gold standard. The purpose of these screening tests that we use in the clinic are with the idea that we have effective treatments and that we're improving the survival time and the life expectancy of those people that are treated. We can also see that when we look at 5-year survival rates, depending on when we classify it by local, regional, distant, or unknown, what percentage of cases are diagnosed in those different stages, and then their corresponding survival rate. So we see the more localized they are within the pancreas itself, the better survival. And as it metastasizes and spreads around the body, we see a later stage of diagnosis and a worse survival time. So here are some more numbers specifically for pancreatic cancer. When we look at the upper left-hand plot, we see on the x-axis the years after initial diagnosis, and on the y-axis we see survival rate. So we see as the years after diagnosis increases, we see a sharp drop off within the first year, year and a half. So most people that are diagnosed with pancreatic cancer are dying within that period of time. If we look at estimated number of incidence cases from 2018 to 2040, again, year is across the x-axis, number of cases is on the y-axis, and we see that we are estimating a steady increase in the number of cancers observed over this period of time. Similar story if we jump right below that, when we're looking at death numbers, we will also expect those to, in the same linear fashion, increase over time. And then if we jump over here in the bottom left-hand corner, again, we're just getting a better idea that most of the diagnoses are happening at a late stage, and most of those late stage cancers then lead to poor survival time. So when we talk about incidence, that's the number of new cases over a period of a specific period, usually a year. And when we talk about prevalence, we're talking about the number of new and existing cases. And with pancreatic cancer, those two numbers are almost the same, meaning that most of those people that are diagnosed within a year are going to end up dying within that same year. All right, so when we think about risk factors, there's several, including smoking, obesity, diabetes, type 2 diabetes, chronic pancreatitis, age, family history, different types of genetic syndromes, gender, and race. So when we take a look at these plots here, it sort of is demonstrating how these factors can vary based on location that you are in the world, smoking rates, high fasting plasma glucose related to type 2 diabetes, and then high body mass index related to obesity. We can see how that varies across the world and depending on where you live. And then as we jump down to the B section of the plot for males and females again, so males is blue, females is the tan color. We can see that there is a different distribution and different intensities. So we see that males will have higher rates of pancreatic cancer, higher rates of smoking. We see that males and females have pretty equivalent fasting glucose, plasma glucose, and we see pretty much the same distribution as we look across age groups when we compare obesity rates with slightly higher rates in the female population. So what percentage of pancreatic cancer can be inherited? So based on some estimates, it ranges from 10 to 15%, with 5 to 15% being represented by familial, meaning it appears like there's some family history, but there isn't unknown genetic link or a genetic mutation within those specific families. And then 70 to 80% is sporadic, meaning that we don't really know the specific causes or a combination of causes that led to pancreatic cancer development within those individuals. There's no family history, there's no specific genes that can be pinpointed or associated with the disease. When we take a look specifically at the genetics a little bit more, so that portion that can be inherited, the most common types of genes that are associated with pancreatic cancer are going to be KRAS, CDNK2A, TB53, and SMAD4. We also have some additional genes that are at lower frequencies, so they don't happen as often within pancreatic cancer cases, and they're not as consistent as the first four. Then there's also, if you look at more of a biological pathway view, there's 12 different key driver pathways and biological processes that have been consistently identified with the development of pancreatic cancer. And the last point here is just a reference I use for that. All right, so if we are comparing one of those genes, KRAS, so here on the y-axis we have KRAS expression, and on the x-axis we are looking at average expression among normal tissue and among pancreas tissue versus tumor tissue. And so we see that the tumor samples that we have show us that there is a much wider distribution and a lot more variation within those tumor tissues compared to the normal samples. We also have a lot less samples that are classified as normal compared to the tumor tissue. But we see a slight variation there. And as we know from our biostats class, the more your sample size increases, the more narrow your confidence intervals will be, and the easier it will be to detect a difference between those two groups. We know that if we would increase the number of normal samples, and as we continue to collect and genotype or sequence tumor tissue, these bars will become more narrow, and we'll have an easier time telling the difference between the mean point in this box plot, the normal box plot, compared to the mean value or the median value within the tumor box plot. So staying with the KRAS mutations that are often observed in pancreatic cancer, again, we see KRAS expression over on the y-axis, and we see different types of copy number categories. So there's a loss in the gene expression. We have multiple copies of the gene, and then we have a gain in expression level and copy numbers. And so we can see that as we move from a loss to a gain, we steadily increase on the amount of KRAS we can find within the samples. We can also see a similar relationship when we look at missense mutations, that as you move from a loss to a gain, we see a steady increase, a linear increase in the amount of expression we can observe. Staying with KRAS gene again, when we look at survival rates, when we have low KRAS expression, so first of all, so we have time in years across the x-axis. We have survival probability across the y-axis. So it's good to be higher in the graph as you move across time. So the blue line is showing us those individuals that have been diagnosed with pancreatic cancer, but have low KRAS expression have a better overall survival over time compared to those pancreatic cancer patients that have high expression of KRAS. And we can see based on the hazard ratio and the log rank p-value that if we're using a p-value cutoff of 0.05, which is standard in epidemiology, would have a significance there. And we see the confidence interval again showing us that it's significantly different than 1. So it's telling us that there's a difference between these survival curves, at least the average of the survival curves. So this plot is a transformed plot. So we take a measurement of tumor purity. So you can imagine that when we're taking biopsies of the tissue, that sometimes there'll be tissue in the sample other than tumor tissue. And so the higher the tumor purity, meaning the higher the sample is of only tumor tissue, we can show that those patients will group together compared to samples that have a bunch of normal tissue, a bunch of blood tissue, a bunch of surrounding tissue aside from just the tumor tissue in that specific sample. And this is important when we are looking at expression, when we're looking at epigenetic factors, because those vary when you move from one tissue to another, when you move from normal tissue to cancer tissue. So the more pure the sample is, the easier time we have at identifying those markers or those alterations within the different data sets. And we can tell the difference between tumor and normal tissue. So this plot is including 3 of those 4 common genes that I showed on a couple slides back, showing the correlation between those genes. So we can read this plot as correlation, negative correlation would be -1, so the darkest blue. Positive correlation would be up to 1, which would be perfect correlation in red. So we see that SMAD4 and TP53 are somewhat related when we're looking at normal tissue. And when we look at tumor tissue, we see a different correlation profile. So that's telling us that within the setting of tumor tissue, the relationship between expression of these genes changes. So that's one way that we could potentially be identifying tumor tissue from a normal tissue by looking at expression of these specific genes. And this is a sort of a visual representation of how different genes might be associated within a specific sample. So if we identify the yellow points are the nodes within these different gene networks, and we see how related they are to other genes that we might have measured within the same samples. And so we see that KRAS is only associated with a few genes in the sample, SMAD4 more, and then TP53 a lot more genes. And that we can see that the correlation between SMAD4 and TP53 is showing up here by connecting lines between the 2 nodes, where we don't have any connecting lines with KRAS, as illustrated in the previous plot. We didn't really see any correlation. All right, so we spoke a little bit now about the 10 to 15% that can be inherited from parents to offspring. But now what about the rest of the 90 to 85% to 90% that isn't covered in the inherited piece? So that's sort of where epigenetics comes in. So genetics is thought of being more, you're born with it, it's static, it doesn't change. And this is more the nature piece to your genetic code. And when we think about epigenetics, we think about mutations or alterations that are affecting the way that our genes are expressed and our genes function within our body. And so most of the epigenetic pieces are thought to be more dynamic. They can be changed by the environment that you're in, the food that you choose to eat, the exercise you choose to do or not do, different behaviors you're choosing to do, maybe smoking, drinking, how are they affecting your overall risk? So all of those things can be picked up, are thought to influence epigenetics, have been shown to influence epigenetic pieces. And so those, even if you don't have a specific mutation or alteration within the gene itself, you can have epigenetic alterations that are influenced by these behavior choices. And those are what impact the overall function of that gene. So is it being expressed? Is it not being expressed? So it's not a specific gene mutation that's influencing that, but instead the transcription is influenced by these different epigenetic features and mechanisms. So the epigenetic piece can explain some of the 5 to 10% that is related to familial, and it can potentially also explain some of the sporadic cases. And the idea here is that your environment can influence genetics and also your epigenetics. So maybe before your baby is born, that's how the environment influenced genetics, how those genes are coming together and forming the baby. And then those genes then can predispose individuals to a specific disease and lead to an overall phenotype. In this case, we're interested in pancreatic cancer. Epigenetics can also be influenced by the environment, but these effects can happen either before a baby's born, or it can be can impact disease phenotypes after, in the choices that as we're growing up and as we're adults, the choices we're making in terms of behaviors. And then again, there are epigenetic factors directly associated with aging, the natural course of aging. And so how does the behavior choices we make, in addition to natural epigenetic alterations with age, leading us to develop a specific phenotype or a specific disease? All right, so when we talk about measuring genetic alterations, we talk about looking at SNPs, copy number variation, mutation within the DNA strand itself. When we talk about epigenetics, we're talking about methylation. We're talking about histone modification. So how is that genetic code or that DNA being wrapped up or unwrapped? So the genetic code, the chromosomes need to be unwrapped in order to allow transcription, and then down the road, translation into proteins happen. And so if those pieces of chromosome, those pieces of DNA are not accessible because they're wrapped around histones, or they're methylated in a certain way that prevents the polymerase from attaching to the DNA strand. That is a few of the mechanisms that can influence whether that gene is expressed or not, or the level that gene is expressed at. And we talked a little bit about how those two pieces come together to form the phenotype. We can also have environmental types of exposures, drugs, pathogens, toxins, nutrition, and how those might come together to also how those three pieces come together to influence disease risk in any phenotypes that an individual develops over their lifetime. All right, so why is it so intriguing for us to take a look at epigenetics in the first place from a from a public health standpoint? So we can think about how we can explain different types of designs and evaluations of interventions that we can develop in more of a biological way. So maybe we can I'll get to that in a second. So there's a potential path to an outcome to overcome genetic susceptibility. So if we can develop epigenetic-based interventions, there's a potential that an overexpressed gene or an underexpressed gene, we can change the level of expression of that gene within a specific organ or within the body. And those therapies are starting to be developed currently in cancer. And then thinking about how epigenetics might represent biologically relevant exposure. So, you know, you potentially can ask somebody how much of bananas they ate over the last week, but there's a piece that we have to think about in terms of how do individuals vary in the way that they're metabolizing that banana. How is their body using the potential nutrients they're getting from that banana? And so if we use epigenetics to try to understand that exposure, rather than set at a granular level or on a biological level, how much is available to specific organs, how much is available to the body, more of a biologically relevant exposure and dose. That's something that's also intriguing moving into the future. In addition to asking people to fill out questionnaires and surveys, we know that there's lots of different types of bias that can occur, whether it's misclassification, so you're shifting some people into different disease categories, whether you're classifying them as diseased or not diseased, or exposed and not exposed based on their answers to a specific questionnaire, or you're looking at more of like a recall issue. So how in depth do people remember what they might have ate, for example? Are they keeping a record of things like that because they're on a diet? Have they thought about that because they've been diagnosed with a disease? So they spend time thinking, well, how could I have potentially developed this disease? What sort of things might I have done in the past that may have led me to get here? So they've spent more time thinking about that than controls. And so that leads to bias as well. But if we can use epigenetic markers to represent those types of exposures, rather than asking questions, can we get more reliable answers? Can we get more valid answers as we move forward and there's more and more of this data available? All right, so just outlining some basic bioinformatics approaches or just one bioinformatics approach that has been taken when we're thinking about analyzing the relationship between some of these omic data sets and cancer. So far, I've talked a little bit about genes and gene expression. I also introduced epigenetics a little bit in terms of methylation and histone modification. A lot of the publicly available databases, especially with cancer, will have methylation markers, data sets. They'll have microRNA data sets. MicroRNA data sets are non-coding RNA that influence the translation of protein coding RNA. So the protein coding RNA, you use that RNA sequence directly to form the proteins that have functions within the cell. The mRNA can interact with those mRNA, messenger RNAs, which code for proteins. and change either the way that the protein itself configures itself within the cell and ultimately change the function of that protein within the cell. Then you can also have DNA mutations, like we talked a little bit about in terms of point mutations, so SNPs, single nucleotide polymorphisms, or you can have mutations happening in terms of copy number, and that also influences expression. And then you go through processing steps. So you run these analyses in the lab. You have to process how good of a job you're doing at detecting different types of pieces of the genomic strand, and then take out any pieces that are outliers. So there's too high of expression, too low of expression. It's not even detected to begin with. So sort of those pre-processing quality control checks with the information coming off the laboratory machines, and then taking a look at exploring the data. And usually these are empirical based approaches like principal component analysis, where you're trying to group a large set of markers, whatever they are, whatever data you're dealing with, into separate groups, and then analyzing those groups as opposed to analyzing each individual marker itself. Trying to do discriminant analysis to further understand which set of markers may be most important in a specific disease setting, and then visualization of your results, trying to understand how those different data types are related to one another and maybe influencing each other within the cell. So we can look at across the entire genome using plots like this. We can look at heat maps, which we'll see a little bit later. And we can also look at correlation plots, which we'll also see a little bit later. So here we can see sort of a workflow. So we're trying to understand how these different genomic data sets can potentially lead to a specific phenotype, in our case, pancreatic cancer. Understanding how different principal components might be changing as we change the size of our data set, how much variance they're able to explain within the data. Are they able to tell us a lot of information? And how exactly are they telling us that information? We can look at correlation types of plots to try to, again, understand how different data sets, different multi-omics data sets are related to one another, looking at those components and those groups of components, understanding in a visual way the entire network of correlations that might be happening, looking for overall patterns depending on the way that we stratify our groups. So cases, controls, are we stratifying them based on stage? Are we stratifying them based on age or a different type of risk factor? And can we identify across a large range of markers a specific pattern to tell the difference between those groups? And then doing some sort of functional analysis. So we've subset it now to different types of genes that are in these different networks. How can we understand what functions might be impacted in a specific disease based on existing pathway information? All right, so thinking a little bit more about pancreatic cancer, it's not just you have cancer, you don't have cancer. This is true with many types of cancers. They're much more complex. And the idea is that the cancer is a dysregulation of what's happening in the normal cell. So here on the left-hand side, we have a normal pancreas, and then we have the setting of the pancreas when this person has pancreatic ductal adenocarcinoma. So we see the environment of the organ itself is dysregulated. It looks like very chaotic, and there's a smattering of different types of cells within this environment. And so this is the idea related to the tumor purity. So if I sampled up here to get my tumor sample, then that would be a pure sample, only representing tumor tissue. But if I sampled somewhere like here, then it's encompassing the matrix, these additional cells. And so that would be lower tumor purity. We can also see that over time, the cancer itself, so the cancer has to start with one cell and then it expands from there. So the environment itself can lead to mutations that developed in the first place. So the altering of one specific cell, then that specific cell has a lineage of cells as it reproduces. So you have clones of that specific cancer cell. But then you also have, because of the environment, created this one cell to mutate itself and start to develop cancer. You have further mutations happening as time goes on, both within the clones from that one initial tumor cell, but you can also have different mutations happening, and each one of those different mutations then lead to additional clones and additional mutations that happen. So as we move from more early on in the tumor environment, where you have more similar types of cancer cells, to as you move down, you have a situation where there's a bunch of different types of tumor cells with different mutations. And so that's why as we move from early stage to late stage, it becomes much more difficult to have an effective treatment, because there's a whole bunch of different tumor tissue cells by then, and only a fraction of those cells will respond to one specific treatment. So that's why a lot of the time there'll be multi types of treatments to try to approach this idea to attack these different lineages of cells and potentially kill off enough to allow the patient to have a longer survival time and potentially cure the cancer. All right, so we can use genomic data sets to identify some of these subsets of cancer types. So this table is sort of highlighting that 4 different techniques were used. Researchers to identify different subtypes of pancreatic cancer. And so they identified different number of subtypes, and they also identified different names for those different subtypes. So we can see in this first column, the number of samples they had to work with and the types of samples they had to work with. That all varies. And so that also then led them to different conclusions from their specific studies. So here's more of a visual overview of those 4 plus an additional 1, showing how some of those different categories overlap with one another, those different subtypes, the different names, and where exactly they're overlapping with each other. So here, the first 2 data sets were using genomic data sets, just looking at gene expression, and then transcriptomics, more of no, I'm sorry, I misspoke. These first 2 are just looking at the DNA sequence, and then these last 3 are looking at the gene expression pieces to understand how we can potentially use that information to subtype different cancers. And the idea there is that if we can subtype them into a specific group, hopefully that will sort of guide which treatments are going to be most effective for that subgroup of cancers. And so we're giving the right treatment to the right subgroup of cancer. All right, so next I'll move on to some of the multi-omics work that we've done at NDSU in collaboration with Mayo Clinic and through funding of the COBRI mechanism here at NDSU, which is funded by the NIH. So for the project, we are working under the assumption that there are a set of components that need to be in place in order for a person to develop pancreatic cancer or pancreatic ductal adenocarcinoma, but that specific set might differ from one person to another. So, we went through an overview of some of the risk factors associated with pancreatic cancer, but not everybody has each one of those risk factors. So everybody potentially has a unique set of factors that leads them to develop cancer. So some of the factors are in common, but some of the factors might not be in common. So the focus was on looking in key genes and pathways to try to understand more deeply how these different types of genomic data sets, whether we're looking at microRNA or gene expression or methylation, how they're interacting with each other, and how they're interacting with some of these common genes that we talked about, like KRAS and SMAD4. And then we wanted to try to identify what sufficient component cause models might be in place for our sample of patients that we're taking a look at. So we'll talk a little bit more about these causal models as we get towards the end of the presentation. But basically, it's setting up trying to identify those sets of factors that are contributing to the onset of cancer within the samples that we're looking at. All right, so we wanted to start first with existing data sets. One of those existing data sets is called the Cancer Genome Atlas, or TGCA. So they have lots of different cancer data sets, including for PDAC, and they've collected information on DNA sequence, gene expression, methylation, microRNA. And so we can look at those samples, analyze that data, try to understand and build these component causal models. We wanted to use several different existing statistical methods and approaches to try to understand how those different approaches influence our results and our unique multi-omics set that we end up observing within our sample group. So here's one of the types of plots that you can take a look at. It's called a Manhattan plot. And so essentially what we're looking at is chromosome number across the x-axis. And on the y-axis, we're looking at a measure of gene expression. Here it's negative log 10 p-value. So here it's telling us which markers across the genome are most significant. I misspoke, it's not gene expression, it's methylation. So which genes, specific gene locations across the genome are most associated or most different when we compare tumor tissue to normal tissue. And so we can see that in general, these first chromosomes are going to be larger. We're going to observe the most markers associated with differential methylation in the early chromosomes because they're larger. We also see that there's a black line indicated here. So when we're doing a lot of different statistical tests, essentially what we're doing is we're comparing each methylation marker across the genome and trying to determine, is there a mean difference or average difference between tumor samples compared to normal samples. And we're running that test for each genomic marker, methylation marker that we're looking at across the genome. So we needed to adjust our p-value for that many tests. So we can no longer use 0.05 as determining whether something's significant or not, but instead we have to divide by the number of tests we're running. So if there can be 450,000 different methylation markers. Actually, with the newer types, there's 850,000 different methylation markers across the genome. And so you're dividing 0.05 by that number to determine what your new significance cutoff should be. So you're adjusting for those multiple comparisons. So we see that there's quite a bit of markers that still remain significant. When we take a look at all of the differentially methylated markers across the genome and compare the average methylation in tumor compared to normal, and we see that the sample size for tumor in this data set is 184 different samples. We have 10 different samples for normal that represent normal tissue. And we see, just looking at the median bars, that there's a pretty significant difference between those two median bars. But again, there's no p-value attached to this test as I presented here. It's more just a visual identification. And so we really want to look more at what relationship we might be viewing or observing for different markers, because some markers will be hypomethylated or have a lower methylation when there's in tumor tissue compared to normal, and some will have hypermethylation, meaning they'll have higher methylation in tumor tissue compared to normal. And so that methylation is directly related to either increased expression or decreased expression. And so it's not always that increased methylation leads to increased expression. It's not always that increased methylation leads to decreased expression. So it's a very individual type of gene relationship. And so that's why it's important to take this general type of summary statistics and actually explore it deeper by looking at those gene network models. All right, so not only did we take a look at modeling one methylation marker and comparing the average methylation in tumor tissue compared to normal tissue, but we also wanted to take a look at, if we looked at high pure tumor tissue compared to normal, if we wanted to look at both low and high added to the same group compared to normal, low purity samples compared to normal, and then high purity tumor samples compared to low purity tumor samples. And so this Venn diagram is showing us how many of those methylation markers came out as significant as we ran the different models. So here, inside this blue circle represents all of the methylation markers that were differentially methylated, comparing high tumor purity compared to normal tissue. Same thing with the green for tumor versus normal, same thing with the orange for tumor, the low purity versus normal, same thing with the purple for high purity versus low purity. So what we're most interested in here is the intersection between all of the different models. So are there methylation markers that can distinguish between tumor and normal, regardless of the quality of the sample that we've collected? That's sort of what this 31 is giving us. So if we look a little bit more into those 31 and generate a heat map. So here on the y-axis are different methylation markers, the 31 different methylation markers. Up here on the x-axis, we see our different stratifications or different grouping variables. This top one is normal versus cases or tumor tissue. The next line is comparing high to low tumor purity. These are again normal, so that directly corresponds to the normal tissue. And then we see unknown purity. And then we see this last one is grouping them into different stages of cancer development. So here we see that there's a pretty clear differentiation between those that are classified as normal tissue and their methylation levels. So blue represents hypomethylation. The stronger the blue, the lower the methylation. The stronger the red, the higher the methylation. Then as we move to the high pure samples, we see that there's a pretty there's a pretty big difference comparing to normal. And then we see that the low purity samples sort of end up in between the normal tissue and the high pure samples. So when I was looking at the diagram, these 31 are sort of can be thought of as a continuous scale. So normal tissue starts here, and as we move up with better purity, we see higher or lower methylation values. And that's just a general pattern. As we can see, if we focus on specific gene or methylation markers, the individual patients vary a little bit as we move across each row. Then we can do the principal component analysis. So can we separate high purity samples from low purity samples, from normal samples using those 31 different methylation markers? And we see that we can definitely separate those high versus those that are low. And we see that the green or the low purity samples sort of overlap a little bit with both the high purity samples and the normal tumor tissue. But it has fairly good separation. If we take a look at biological processes and functions that are represented in this set of 31 different methylation markers, we see that they're associated with these different functions and this one pathway is identified. So the idea again is to try to understand a little bit more about what these methylation markers might be doing, because a lot of their functions aren't necessarily known. And also as we connect it to other genomic data sets like gene expression or microRNA, how are we seeing these similar biological processes or functions overlap between those different data sets? All right, so now this Manhattan plot is showing us gene expression data. Here we see that there's a lot less significant genes that are identified as differentially expressed in tumor compared to normal tissue. And that is partly a function of there being less genes in our set compared to the number of methylation markers we are looking at. The genes that we have to analyze are closer to around 3,000 to 5,000 compared to the methylation markers, which were more like 450,000. Then we can look at that volcano plot where we're looking at the downregulated genes and the upregulated genes, and we see that there's a lot more downregulated genes in pancreatic cancer. These heat maps are slightly different. So across the x-axis here, we have different genes, and across the y-axis here, or the rows, we're showing different microRNA markers. And so we can see overall, if you just take a look at the entire plot, this top one is showing us a normal situation. This bottom one is showing us the tumor situation. And we see that there's a attenuation of expression of microRNA and genes, an attenuation of the correlation between those two omic sets in the tumor situation compared to the normal situation. So we see much deeper colors in the top plot compared to the bottom plot, telling us that there's a dysregulation of the normal correlation structure and the normal regulatory processes in the situation when we have cancer compared to the normal situation. When we look at just the gene expression data and take a look at principal component plots, we don't see as strong of a separation between those three different groups, the high purity, the low purity, and the normal tissue. And then when we look at the different biological processes, functions, and pathways, we see that slightly different items are getting highlighted with this gene set, sort of emphasizing that it's important to take a look at the different multi-omics data sets, because you get different representation and different interpretations of what might be going wrong within the cell. All right, now so, focusing on the microRNA and taking a look at downregulated, upregulated genes. So we see that all of the mRNA markers that we found differentially expressed are all associated with downregulation within the tumor environment. And then this is just, again, showing the genes and the microRNA, showing that the correlation breaks down in the setting of tumor compared to the normal situation. And then as we look at microRNA, there's really no ability to distinguish between those different tumor and normal groups. And that's because the microRNA, one microRNA interacts with a lot of different genes. So we wouldn't expect it to be able to distinguish tumor from normal very well, because it's involved with so many different types of genes. All right, so from the analysis we've completed so far, some important points are that single genomic analysis association studies can result in many false positive associations. So one way we tried to avoid that in this study was, one, to adjust for the multiple comparisons, two, to take a look at the different, to do that Venn diagram and identify those markers that are only associated with all the different models that we're looking at, to give us the stronger, potentially give us the strongest predictors of distinguishing those tumor cells from those normal cells. Cancer is very complex, as we've talked about already. And so trying to understand how these different genomic pieces are working together to lead to the onset and progression of cancer is going to be very important. So combining those different data sets together, working across different data sets, so publicly available data, taking a look at potentially in the laboratory, looking at cell lines and animal models, looking at patient tumor samples that have been collected in the clinic, could potentially allow us to remove some of the noise in the data and remove further some of those false positive associations. And again, just wanted to highlight that pancreatic cancer tumors are heterogeneous, but if we are looking at average genomic values within tumor samples, we're likely to identify those alterations that happen early in the disease process. So that if we're comparing within the pancreas, if we're comparing tumor compared to adjacent normal tissues, we're going to be highlighting those alterations that are likely to have happened early on, because we're just taking an average for our entire sample. And then in this project, we're interested in, again, trying to have a deeper understanding of how some of these markers might be related to genes that we know are associated with pancreatic cancer, like KRAS and SMAD4, and how are these genes mutated. In the absence of mutations, are we seeing epigenetic alterations happening or affecting the gene expression in a tumor environment? All right, and then building these specific component cause models. So the idea is that there's a set of factors that contributed to the development of pancreatic cancer within an individual, and that this set of markers is likely to differ from one individual to another. There could be overlapping causes in both. If that cause is in every single causal model, that's telling us that it is a necessary cause. But we already know in the setting of cancer, unless we're able to subtype it enough, we are not going to have any factors that are going to be represented in every causal model. We can have, so each one of the individual risk factors here identified by the letter is called a component cause. And then as an individual develops a series or a set of component causes, the minimal set of component causes that leads the person to develop disease is called the sufficient set. So that's a set of minimal risk factors that person had to develop or had to have in order to develop their disease. In this situation, pancreatic cancer. So with this work, the idea is to try to subcategorize pancreatic cancer enough so that we can get some overlap in causes, identify if there's any necessary causes, and also identify those genomic markers that lead a person, that likely led a person to develop disease in the first place. All right, so I just want to acknowledge my team here at NDSU, and in this slide, a couple of my students are left off. David, Nijam, and were left off this slide, and they're PhD students in the Genomic and Bioinformatics program. I received funding, both a seed grant and also a junior investigator level funding in the COBRA project here at NDSU, collaborating with Mayo Clinic for analyzing their samples they've collected, and then working with a computer scientist at the University of Wisconsin. All right, that is the end of my slideshow. Are there any questions?
Thank you very much. Sorry, my microphone wasn't working last week. Can you hear me?
Yes.
Okay, perfect. Thank you so much for that. Really interesting. I know more about pancreatic cancer now than I ever have, and I have a couple of questions, but I'm going to open it up to our participants. I think we're a small enough group that if you feel comfortable, feel free to unmute yourself and provide your question. Otherwise, if not, feel free to put it in the chat box. Okay, so while people are thinking of their questions, I do have a question. I mean, this is obviously very complicated. The work you're doing is extremely complicated. I'm not going to pretend that I understood everything that you said today. But my question is, so knowing what we know now about pancreatic cancer, and knowing what you know now about pancreatic cancer, there are a lot of these programs like through Stanford where you can do genetic testing to see if you're more predisposed to certain types of cancers or certain conditions. Is your area of study one that lends itself to that type of testing, that type of research, where you would be able to pinpoint to a greater extent the type of tests that somebody would need to undergo to determine if they were more predisposed to certain types of pancreatic cancer?
Yeah, that's definitely the way that the field is moving. And one of the objectives of doing this type of genomic analysis is that you can create a set of biomarkers to create that panel that can sort of tell you whether you are at risk of getting a specific disease or not. So it's still pretty early on. And so at least with pancreatic cancer, there isn't well-established biomarkers or panels that have been set up to this point yet. We're definitely getting in that direction. But part of the problem is that most of the cancers in general are pretty rare, and pancreatic cancer especially. So how are you going to choose who in the population is going to get screened for a specific cancer and who's not? Can you develop a biomarker panel that could sort of identify all of the different cancers? And so maybe that would be more relevant for everyone to take than sort of this cancer specific type of panel, maybe as a first step. And so those are some of the challenges that we run into when we're trying to screen for cancer. Is that it's relatively rare. How are you going to identify those individuals that are most at risk? And how are you going to ensure that you're identifying those people at an earlier point and have effective treatments that can then be used within that population to extend their life and hopefully ensure that they have a high quality life?
Thank you. Mark. Dr.
Well,
Strand.
as a follow-up to that, would it also be the case that given that there's only 10 to 15% of pancreatic cancers are believed to have a genetic, underlying genetic cause, that also makes it a little bit like a needle in a haystack experiment? And maybe up to 20% if you add in the familial, but not yet demonstrated to be genetic. Is that also a factor?
Yeah. So that is also going to be difficult if you're just focused on the gene expression piece. But if you look at some of the epigenetic markers, you will have you increase your chances of identifying those people that are likely to have cancer, even though it's not detectable at this point. The other, you reminded me that the other difficulty with doing these types of panels is that a lot of the panels and a lot of the data that's been collected is at the tumor level. So that, for understanding biological functions and the importance, what might be going on in the tumor itself, those are important. And those panels can be used once an individual is diagnosed to try to manage the treatment plan. If we want to try to screen, there has to be a less invasive way to do it. We can't biopsy everybody's pancreas. So we have to look in the blood. And so the challenge there is how can you identify those blood-based markers that may correlate with the cancer markers that are relevant, functionally relevant, but don't necessarily represent anything related to the disease, but maybe represent some sort of immune body-based response to the existence of cancer. And so that's another challenge that we face with many of these different types of cancers.
Yeah, thanks.
So Dr. Jansen, I have another question, and I'm not sure if you can answer this one, but cancer research has been ongoing for several decades. And I'm not an expert in cancer research whatsoever, but are we in the infancy of pancreatic cancer research, or the type of research that you're doing as compared to, let's say, breast cancer or lung cancer? You know, how would you compare where we are in the evolution of the sciences behind detecting pancreatic cancer?
Yeah, so in cancer genomics in general, we've known the most about genes, DNA sequence, and DNA expression, gene expression for the longest period of time. And as we continue to develop laboratory techniques and learn more about processes that are going on in the cell, the epigenetic pieces were identified maybe in the mid 2000s, like 2005. Before that, gene expression maybe in the mid 90s. And so as we learn more about the way that these elements are interacting with each other in the cell and how they go on to influence biological processes and pathways and function of the cell, we learn more about those things. But so the cancer genetics has been around for the longest period of time, and that's been around for all types of cancers. And then as we move to present day, we've learned more about those epigenetic factors. And again, those are most intriguing to us in public health because it's a way that we can potentially modify our genes through behavior modification, through targeted treatments, maybe in the future. And so those pieces are really at the early stages.
Thank you.
Mm-hmm.
Any other questions that we have?
Oh, yeah. Why are you anticipating such a substantial estimated increase in pancreatic cancer up until 2040? Is it just a chronological trend of aging, global aging, or is there another factor?
Yeah, I think that probably a combination of risk factors, the increase in the risk factors, especially things like obesity over time, contribute to that. Type 2 diabetes over time contributing to that. And maybe our overall treatment and keeping people healthy for other types of diseases. And so, people developing pancreatic cancer because they're living longer, as opposed to maybe necessarily the incidence rising.
Mm-hmm.
But probably a combination of both.
You mentioned obesity, and I'm wondering if you have identified some of the specific epigenetic factors that are considered with pancreatic cancers. One thing I thought of was gestational diabetes potentially because of the organ association, but are there known epigenetic factors, behaviors, risks, exposures that are candidates for these impacts on the genetic expression?
Yeah, and so it's still really early. So, and the number of samples that we have to analyze is pretty minimal. So it's difficult to try to identify those, but there are definitely epigenetic features that are related or have been identified in maybe a set of obese individuals, maybe a set of type 2 diabetes. And so there is some overlap between those. But again, probably if you look at epigenetic markers, what you'll identify is that for certain people that maybe have a diagnosis of these things, those markers will show up as in their panel as differentially expressed. But if you look at those people that don't have obesity or type 2 diabetes, those markers won't show up. So when you take an average of a group of people, depending on how many have obesity, how many are obese and how many have type 2 diabetes. That sort of influences whether you're going to identify those markers or not.
Do we have any other questions? Okay, I'd like to thank Dr. Rick Jansen for sharing his information with us today. Really appreciate it. There's a lot of really great information. And if anybody has any other questions, could they contact you directly, Dr. Jansen?
Yeah, that'd be great.
Wonderful.